Hello friends, myself Dr. N.Bhale, Professor Department of Botany, Art Science, Commerce College Nadu, Taluka Tuzapur District Usmanabad, affiliated to Dr. N.Bhale University, Aurangabad. In my last terminology video, I would discuss the different terms related with the work of mycorrhizal fungi. Here also I am discussing about the mycorrhizal fungi and its important terminology, part 2. This is a composite spore wall. Composite spore wall is nothing but the total wall of a spore consisting of all wall groups and wall types is called a composite spore wall. Collar, a term applied to the pedicel between the spore and hyphae in Ectomycorrhizal species. The term is used especially if the pedicel is extremely short. So let us see now here. This is the Ectomycorrhizal species, and this Ectomycorrhizal species, this mark is showing the collar of our pedicel of this Ectomycorrhizal species. Then coriaceous type of wall layer. One of the wall types occurring in spores. It is a robust, tough, flexible wall becoming leather-like in appearance in hypertrophic solution. When the spores dipped in hypertrophic solution, so that time it's found the flexible, tough spores. So let us see now here. We have sketched the diagram on the basis of our study. So this is a coriaceous type of wall layer are present in mycorrhizal fungal study. The next term is there is a cyanophilus. Cyanophilus is nothing but is a readily absorbing or stained by Netian blue dyes. This is one type of dye or the stain are there, and when they are absorbing such type of the stain is called a cyanophilus type of cyanophilus. Second is a dark septate endophyte, that is a DSE. Such kind of infection are thoroughly or frequently found in the root colonization, that is called the dark septate endophytes. Is nothing but it is a plant root colonizing fungi that are often dark pigmented with septate hyphae and forms structures inside plant root cells. So this is the dark septate endophytes. Let's see now here. These are the dark, thick-walled root colonized cells are there. So this marked portion is showing the dark septate endophytes. Then dextrinoid, that is staining dark red with Melzer's reagent, indicating a low amount or absence of starch 1 compound. So this phenomenon is called as a dextrinoid. Next, digitate. Digitate is having blunt finger-like projection is called as a digitate. Then icunulate is a type of the spores. That is, spores are the surface with finely pointed spines is called as a icunulate. So let's see now here. We have sketched the diagram. So this is the finely pointed spines are found on the surface of the spores. So this is called as a icunulate type of the projections. Next, this is the Ectomycorrhizal. Ectomycorrhizal is a mycorrhiza with hyphae that do not penetrate the cells. Then Endomycorrhizal, mycorrhiza in which the fungal hyphae penetrates cell walls of host plant. Then Endomycorrhiza, there is nothing but is a mycorrhiza showing characteristics of both ecto and endomycorrhiza. This is called the ectoendomycorrhiza. Then endophytes, that is a fungus living within the plants, often without causing visible symptoms. This phenomenon is called as endophytes. Then advancement wall, one of the wall types occurring in spores, spore wall that frequently deteriorates readily and having a short existence. Then expanding walls. This advancement wall is look like the structure that is known as the expanding wall. Then one of the wall types occurring in spores, spore wall distends and increases in thickness when treated with lactic acid containing compounds, that is called as polyvinyl lactoglycerol or polyvinyl alcohol. So this is called a PVLG. So this is one of the expanding wall, and this expanding wall structure you might have seen. This is one type of the wall layer or ornamentation. Next, extra radical or internal external hyphae, that is soil hyphae. It is a filamentous structure which comprises the fungal thallus body in the soil. It acquires nutrients, propagate the associations, produce the spores and other structures. So let's see now here. Already I have discussed about the extra radical or external hyphae. So this is the ERS or ERH are there. That is known as extra radical hyphae. Now here germinal wall or germinal wall is nothing but is the innermost wall of, especially in the Gigaspora species, on which papillae are born and found which germ tubes arise. So here such type of germinal walls are there in this wall ornamentation, and especially such found in Gigaspora species, that is innermost wall. Germination seal, there is a germinal seeds are there, seal, an oval or to irregularly shaped structure from which germ tubes arise from the inner membranous wall and especially present in the Scutellospora. So let us see now here. So this is the Scutellospora spores, and in the inside, so you might have observed the germination seal. So this is seal like structure, it appears in the spores of Scutellospora. Then glomalin is the very most important things about the presence. It is a protein secreted by arbuscular mycorrhizal fungi into soil for aggregation as well as the compact. So this is very most important proteins are present, that is known as glomalin. Then harding net is nothing but the network of hyphae found intercellular between the root cortical cells. So this is the terminology. Then hostorium. Hostorium is nothing but very most important parts for the absorption of food materials from the host. The structure usually a modified root connecting a parasite to its host. It physically penetrates the host and function by connecting its vesicular tissue to the parasite. Then hypal attachment or substanding hyphae. Substanding hyphae or hypal attachment is very most important parts of the attachment along with the spores. The hyphae emanating from a spore or other structure from which spores arose. So let's see now here. So this is spores, and the spores connecting the hypal attachment, and this hypal attachment is nothing but it is known as substanding hyphae. So these are the different two spores. This is substanding hyphae. Here also the mark showing the substanding hyphae. Then hypal terminus, nothing but is a sporiferous saccule. It's called a saccule. That's the sporiferous saccule is nothing but is a specialized swollen hypal tip from which attached tapered hypha bear spores laterally, especially in the genus of Achelospora, as well as internally by the enterospora. And the structure here, let us see now here in this figure. So this is the structure it found in the Achelospora, and this structure, that is the uppermost structure, is called as the sporiferous saccule or hypal terminus. So this is the terminology related to this mycorrhizal fungi. Then internal hyphae or intraradical hyphae, hyphae which grow within the cortex of a root to form a colony and later develop arbuscles and vesicles, especially the internal hyphae. So these are the arbuscles are forming internally. So these are arbuscles, as well as they are forming the hypal colonization, as well as they are forming the vesicle. Means everything they are arbuscles, vesicles and hypals are forming in one colonization, and such phenomena is called the internal hyphae or intraradical hyphae, as well as they are forming arbuscles and vesicles. Next, inoculum is very most important aspects because it is symbiotic association of this mycorrhizal fungi. No such cultures are formed. So by taking the support of some restoring plant species to prepare the inoculum, and the inoculum is nothing but the substance used to make an inoculation resting spores, hypal fragments, hyphae along with the arbuscles, vesicles in living roots, which is extracted from soil and prepared the inoculum. So this is called as inoculum. Next, this is a laminated wall. Laminated wall is nothing but one of the wall types occurring in spores, a many layered wall with layers increasing with age, and this wall is known as a laminated wall. So this is the spores are having many layered or forming, and such wall or nothing but is the ornamental wall is called a laminated wall. So this is all about the terminology in this video. So please watch this video. If not subscribe my channel, please subscribe, share, like, and send to others. Thank you very much.